Okay, so we better make a start. So hello everyone, thank you so much for registering for this webinar and for turning up. So my name is Denise Carvalho Silva, and I'm the scientific outreach lead within OpenTarget. So my role is to be a bridge between the developers and bioinformaticians in the team and our user communities. So I try to translate what they are doing to you guys, and also I feedback them with the suggestions and ideas that you may have. So today, as part of this EMBL-EBI webinar series, I'm gonna tell you how you can use our OpenTargets platform to find target and disease associations for improved drug target identification. So the goals for this webinar is to tell you what the OpenTargets Consortium is, and then to focus on the OpenTargets platform, which is this tool that can allow you to find target and disease associations. And I will also I'll go through some live demos to tell you and show you how you can navigate the platform. So the outline, I will start off with an introduction, especially telling you guys what the consortium is, and then focusing a little bit more on the platform. I will then go through the live demos. I've got 3 demos there for you, and then I'll wrap up the course towards the end. Okay, so if you are working in drug discovery, the chances are that you have already come across with a cartoon similar to this. So what we are trying to show here is the timeline of a drug discovery path. Before I joined OpenTargets, which has been a year now, I was completely unaware how long it can take from the very first step of my drug discovery journey all the way to the end, when the drug is actually marketed and available for the patient. So this can take up to 20 years, given or taken. So we start off with an idea, we then go on through some basic research experiments. You will probably be doing some experiments using animal models, whether they are rodents, mice, or rats, or probably zebrafish. Then you move on into the clinical trials, which usually is the most expensive part of this timeline. The clinical trials are divided according to different phases. Then, if all goes well, you end up having your drug approved by the FDA, for example, and then you are ready to have your drug targeted and used for the treatment of the disease you're interested in. So this is the timeline of a drug discovery path. However, as some of you may guess, there are several challenges along the way. So some of the challenges are, this path is very lengthy. As I said, it can take up to 20 years. The path is very costly, especially when it comes to that stage of clinical trials. So you are dealing with patients or with healthy volunteers. So that's extremely expensive. And unfortunately, there is a huge there is a low success rate in your drug discovery journey, which means that there is a high attrition rate. So for attrition rate, you can think of the number of dropouts. So the success rate varies depending on the therapeutic area that you are interested in. So let's suppose if I'm interested in cardiovascular disease, on average, the percentage of success is around 20%. So, and that's one of the highest success rate in drug discovery. But on average, and across several different therapeutic areas, the rate of success is pretty low. So those are some of the challenges. And that's when open targets comes in. So we would like to change these attrition rates. So we would like to make this journey more successful. And if we were to fail, we rather fail earlier. So we rather fail in the basic research or on the idea that we may have, rather than fail towards the end of the timeline. So Open Targets is a partnership between academia and industry. From the academia side of things, we've got EMBL-EBI, which is where I'm based, and also the Wellcome Trust Sanger Institute. So both institutes are located on the same campus here in Kingston, just outside Cambridge in the UK. Our industry partners are GSK and Biogen. And what we want to do, we want to change the way that drug discovery is performed, and we will do so by allowing the systematic identification and selection of targets. And we are very much interested in the very early stages of that drug discovery timeline. So we got this bold goal of being the world leader for human target discovery. And this is the virtual cycle that we are very much based on. So we want to support the decision making by people working in the industry or academia when it comes to drug discovery, by allowing the selection of drug targets to be done based on the data that is out there. We are very much committed to sharing all the tools and analysis and data that we create. We want to publish this data and the tools, and everything will be open for everyone. As I said, we do want to change those attrition rates. So we want to allow for that drug discovery timeline to be more successful. And we are very much interested in finding what causes, which pathways, for example, are affected and perhaps leading to a given disease. So within Open Targets, there are two major areas of work. On one side of the consortium, we've got the experimental projects being carried out in wet labs. So this is mainly carried out at the Sanger and GSK, but also Biogen. And we are interested in three major therapeutic areas. So we are interested in oncology, neuroscience, and also immunology. So on this aspect of the project, we are creating new pieces of evidence so that we can link a target or a gene to a disease. And in order to create these new pieces of evidence, we rely on state-of-art technologies. So we are dealing with CRISPR-Cas9 editing. We are also exploring disease models in humans. So we are going away from mouse models or zebrafish models. So we are using organoids and iPS cells as cellular models for the disease. So this is the experimental side of things, creating new pieces of evidence. On the other side of the coin, within Open Targets, we've got what is known as the core bioinformatics pipeline. So what the team has been done, we have been integrating the currently available data out there into a place that can be used by anyone, everywhere, so that you can try to find possible genes associated with your disease of interest. So this integration has been done by my colleagues, and it's available through our web interface, and we also have additional and alternative ways programmatically that you can get hold of this data. So those two areas, those two independent areas of Open Targets, they are concurrent, they are working side by side, and I strongly recommend you to have a look at our projects page to get more information regarding those two major areas of work. So from now on, I will focus on the core bioinformatics pipeline side of things, especially by introducing you guys to our platform. So the Open Targets platform was developed and it is maintained by the core team here at EMBL-EBI. And our goal is to allow our users to identify target and disease associations. And one of the unique aspects of the Open Targets platform is that everything that is, all changes and all data that we've got in there, everything is very much driven by you, so by our user communities. The first release of this platform took place in December 2015, so it's a very recent addition to the portfolio of web interfaces or databases within the API. So based on that, I will try to launch a poll now, just to have an idea if you guys have used the Open Targets platform before. So I'll launch the poll right now. So you should be able to see this quick poll on your desktop. So if you could just say yes or no, that will be very useful. So I've got 40% of you voted, 60. Okay, so around, yeah, 1/3 of you guys have used the platform before, so hopefully you'll be learning something new today here. But the majority has never used the Open Targets platform before. So don't worry, this will be an introduction and you will always be able to get back to the team with ideas and suggestions, or if you are stuck with the platform. So I'll close the poll now. You should all be able to see the slides again. So here we are. Okay, so yeah, thank you very much for answering that first poll. So one of the bedrocks of the Open Targets platform is defining a way to classify diseases. So again, the platform is a tool that allows you to find associations between diseases and genes. And we use genes as proxies for our drug targets. So focus on the disease first. We need the diseases to be classified and organized in a way following an ontology. So some of you may or may not have heard the word before, ontology. You can think of it as a smart dictionary of relationships between entities. So in this case, it's a smart dictionary of relationships between diseases. The ontology of choice when it comes to diseases for Open Targets is the EFO. EFO stands for Experimental Factor Ontology, and it was created at the ABI and it's maintained by the ABI. So it's a way to organize diseases. It follows a controlled vocabulary, and it also provides a hierarchy, which gives us an idea of the relationships between diseases. So here I am showing, for example, asthma. So let's suppose I'm interested in asthma, and I know from this ontology that asthma is a child term of respiratory system disease. Whereas I can also find out from this ontology that there are perhaps one subtype of asthma or perhaps more. In this case, I've got a childhood onset asthma. So this gives you the hierarchy of the disease, and it shows you the relationship between my disease of interest, its parent terms, and its children terms. So there are several advantages of relying on an ontology. So it allows us to achieve consistency, and it also increases the richness of the annotation, and it allows for easier and automatic integration, which is everything that the Open Targets platform is about. And in terms of consistency, you can think the same disease can be spelled differently. For example, if we are looking at the US spelling or the British spelling, so that can create inconsistencies. And by having an ontology, we overcome some of those problems there. So that is our disease, which is classified following the EFO. But we also now, on the other side of our model, we need to focus on the targets or genes. So we use the ensemble gene IDs as a proxy for our targets. And in order to link a target with a disease, we very much rely on evidence, on biological evidence that is available out there. So this cartoon shows the original set of evidence that we started with back in 2015. And more are being added as I speak. So currently, these are the toolkit of data sources that we rely on. So they are all publicly available, and they can belong to databases here, the EBI, or they can be also in other places as well. And our data types, they can be divided according to those major fields here. So we look into data classified as germline, so genetic variants that are germline. We also look at variants that are somatic, so those somatic mutations, they are very much implicated in cancer, for example. So this is when it comes to the genetic types of evidence. We also rely very heavily on drug information. Currently, this comes from Campbell only. We look at affected pathways. We look at whether or not the gene has the expression of this gene varies between diseases and healthy tissues. We also look into animal models as a piece of evidence to make the association between target and disease. And finally, we also mine the scientific literature, looking for the co-occurrence of the target name and the disease name in the same sentence. So this is the tool set or the toolkit of data sources or data types that we have in the Open Targets platform. And for each of those data types can be divided into different data sources. And this number is very much increasing. And we would also like to hear from you whether or not we should add a given data source. So just let us know. So this is the present and future of the Open Targets platform here. So we perform the data integration of data sets that are publicly available. So some of those icons here you may have seen before, you may be familiar with them. So we've got data from the GWAS catalog, Expression, Atlas. We mine the papers through Europe PMC. We've got data coming from the European Variation Archive, COSMIC, which is based at the Sanger Institute. We also have the Genomics England data. So if you guys are based here in the UK, you may have heard of this consortium. And also, we also have FIWAS data. So the Open Targets platform is very unique by providing all those different data sets in one place. And just a reminder, those different data sets, they are used as pieces of evidence to make the association between target and a disease. So this is the present. So this is how the platform looks like today. But the future, and hopefully the not very distant future, through our data generation. So you may remember we've got also the experimental side of things. So our colleagues working in the lab, in the wet lab, so they are creating new data from the oncology point of view, immunology, and also working on Alzheimer's and Parkinson's, to create new data so that it can be fed into the platform, so that you can view them in a very easy to use information. So I've got just one Argo or Geoterms also integrated. I think there may be a question there. I'll refer to them later if you don't mind. But yeah, we do have gene ontology and disease ontology as well integrated in the platform. Yeah, but we can look into that later. Okay, so, and this is just a sneaky preview how the data in the platform could look like. So before I move on into our live demos, I will just mention very briefly how we score or how confident we are with the associations. So for every single piece of evidence coming from those disparate data sets or data sources, for every single piece of evidence, we compute a score. So this score is dependent on the evidence, so it's and the factors that contribute to the score vary depending on the evidence that I'm working on. So don't worry too much about the details, but it's important to mention that we look at the evidence, we calculate a score for the evidence. So let's suppose I've got one SNP coming from the GWAS catalog, and that SNP will have a score which is computed by us. Then, so this is the first level of scoring at the evidence level. Then we combine every single SNP coming from the GWAS catalog, and we use this, we use a mathematical formula that's known as harmonic sum, and aggregate every single data coming from the GWAS catalog. And we do so to provide a second level of score, which is what is known as a data type score. So the score coming from the GWAS catalog, we've got score coming from UniProt, a score coming from PheWAS, and so on and so forth. So this is the second level of scoring. Then all those similar data types here, they will be combined again using this mathematical formula. They will be aggregated and they will give rise to the genetic association score. This is the third level of scoring within the platform. And finally, all those individual data type scores will be combined, again, will be aggregated using the same formula, and this aggregation will give rise to the overall association score. So this is just a brief overview how we go about giving you some idea of confidence on our associations. So based on that, I will move on to the live demos. There are a couple of workflows that you can explore with the platform. So you can start by looking at a disease, so you can search for your disease of interest. You can also search for your gene of interest. We also allow for drug names and phenotype to be searched as well. So if you've got a phenotype or a drug, you can also pop that into our search box, and those drug names and phenotypes, they will be matched to either targets or disease. But for the interest of time, so let's focus on this disease-centric workflow. So let's suppose that I'm working on multiple sclerosis, and I would like to know which information Open Targets platform has got out there for me, such as which targets are associated with that disease. So the first thing I need to do is to choose my favorite internet browser. Those are the ones, listed here, those are the ones that we support. So if you like Internet Explorer, make sure that you use version 11 or above. So in my case, I really prefer Google Chrome. So I will bring up my Google Chrome here. So here we are. And you just go and search for targetvalidation.org and you will see our homepage there. So it's a very, there's not much here that you can miss in terms of a search box, very similar to Google. There is plenty of information here that you can explore when you have time, including plenty of help and some stats as well, based on our latest release. So since I'm interested in multiple sclerosis, I will type in multiple sclerosis there, and here we are. So I can either hit enter from my keyboard, or I can just select the top hit here. So this is what we call the associations page for multiple sclerosis. So what we are doing here, we are showing this heat map, and we are ranking and scoring all the different targets based on the current set of evidence and data sources that allow us to associate them with multiple sclerosis. So based on the current version of the platform, we've got just under 3,000 targets associated with multiple sclerosis. So you are more than welcome to explore this page. So you can see, if you scroll up and down, you will see on the left-hand side there are different ways that you can filter this data by. So for example, let's suppose you're interested just on the targets that we've got genetic information for. So you just tick that box over here, and you will see now the number of targets has reduced from 3,000 to 300. So it's a more manageable number of targets that perhaps you want to prioritize. And again, if you expand this version or this arrow here, again, we list all the individual data sources that make up this data type. And again, you can pick and choose whichever data source you are interested in, or the data source that you trust the most. In addition to the data type, you can also focus perhaps in specific pathways that those genes will be involved with. You can also focus on specific target classes. So let's say those that are a membrane receptor, or those that are ion channels. So again, this can have an impact on your drug discovery. And also, if you've got a list of targets that you know they are already implicated in multiple sclerosis, you can simply upload that list of targets here, so you can filter the associations of bioper targets according to the list of genes that you are already working on in multiple sclerosis. So lots of things here that you can filter by. So I will just remove this filter here. I'm back to the default page. So here we show this table, which very much is a heat map. Again, if you scroll down, you can see there are different shades of blue, darker blue there. So if you go across the different pages here, again, you see something very light blue, medium blue, and a very dark blue here. So those different shades of blue, they are telling you, they give you an indication of the scoring framework that we apply. So the lighter the blue, the weaker the association is. This table can also be sorted. So by default, it is sorted by the overall association score. But again, you can play around here with the different arrows and followed this second column here, which is the overall association score. We also provide individual scores for each of those data types there. So again, you can pick and choose whether, for example, you may want to focus on the target that has got the top score when it comes to genetic association for multiple sclerosis. So there's a lot of things here that you can do. And you can also download this table if you want to as a CSV format. Okay, so this is just a brief overview of our associations page when you start from this disease-centric point of view. So now this will lead me to the second demo. So you did find there are thousands and thousands of targets associated with multiple sclerosis. The associations have been done, rely on different types of evidence, genetic evidence, drug information, or mining the literature. But let's suppose that now I'm interested in drilling down, delving deeper on the exact evidence that has been used for a given association. So let's suppose that I'm interested in the CD86 gene, and I want to find out what Open Targets has got in store for this association. So again, I'll move on back to my page. So here we are. So what you can do from this page, you can simply search for CD86, and here we are. So that filters, again, that huge number of targets down to the only one that I'm interested in. So it gives a summary. Yes, I've got a pretty strong score, overall score. The strongest actually comes from genetic information, and I've got some drug information there as well, some expression data, and also a few papers have been mined as well, highlighting the association between this target and that disease. So in order to delve deeper in the evidence, so I can click in any of those cells, and this will take me now to what we call the evidence page. So the evidence page is characterized by this flower, and this flower is just a brief overview of the data which is available and has been used by Open Targets to compute this association here. So we've got some genetic information, drugs, text mining, and RNA expression. Things that are grayed out simply show that we don't have data yet for those data types. So from here you can explore a bit further. If you open up those tabs, so you will see the exact data that we have used for the association. So we list the variants here. We tell you where those variants come from, in this case the GWAS catalog, and we also point to the original paper that has been curated by the GWAS catalog curators. So, and from here you can explore and you can go to external sources as well. In this case you can go to Ensemble. So this gives you an overview of the different SNPs that have allowed us to associate CD86 with multiple sclerosis. And again, you can download this table if you wish. In addition to this dry table, we also provide this cool interface here. So basically this is as we call it, like a mini browser. You may be familiar with some genome browsers out there, such as Ensemble. So this is a mini version of a browser. So we simply show here the gene itself, my gene of interest, in this case CD86, all the possible alternatively spliced transcripts. Again, if you're not too sure what the different colors mean here, just have a look at the legend. And we show as lollipops all those variants that map to this region of the human genome. So this gene is located on chromosome 3. Here we've got the coordinates. And again, you can scroll down or scroll upstream of this gene. You can zoom in and you can zoom out as well if you want to. So it's very interactive. And the lollipops, as I said, they are the actual variants that seem to be associated with the disease of interest. So in this case here, again, the different colors of those lollipops, they will mean different things. In red, those are mutations or variants in CD86, and they are very much associated with multiple sclerosis. However, the variants in green, it's a variant very much mapped to that region of CD86, but that variant is not associated with multiple sclerosis. It's rather associated with something else, with another disease. So those colors, they do mean, they do have a meaning there. So it's worth you guys exploring this if you want to. So that's the evidence from the genetic point of view. And as I said, you can refer back to the provenance of the data. You can, if you're not too sure, if you don't trust this, the curation from GWAS catalog, or you don't trust what OpenTarget is showing here, we do link you back to the provenance of the data that you can make your own judgment call. So in addition to drugs, to genetic associations, we also have drugs. We've got drug information coming from Campbell, and this allows us, again, to associate a disease with a target. So we've got one drug only. Those drugs from Campbell, we know the mechanism of action for those drugs, and we also, those drugs, they are either already marketed or they are currently in clinical trials. So again, this information can be useful for you. And again, as always, we link you guys back to the provenance of the data, so you can go to the clinicaltrials.gov and explore and find out more about this clinical trial, which is currently in phase 2 for this drug in patients with multiple sclerosis. So there is a lot here that you can explore, including the expression data coming from Expression Atlas and our mining the scientific literature. So we are looking for the co-occurrence of my target name and that disease. So again, that can give you an idea of the papers out there where that association has been observed. And again, you can drill down a bit deeper if you wish to do so. So from this page, there is plenty more you can explore. For example, you can find out a little bit more about the disease itself. So I will go and open this up in a different tab. This is what we call our disease page. It's a profile of the disease. So it gives you a summary or a definition of the disease, providing this definition is known. It also gives you all the known synonyms, and again, you can search for any of those synonyms and we should take you to the right place. We list you all the different phenotypes linked to that disease, and again, you can search for those phenotypes if you want to. We also list every single drug from Campbell that are in clinical trials targeting different genes, but using patients with that condition. So either multiple sclerosis or subtypes of multiple sclerosis. So we also take into account the children terms of multiple sclerosis. And then if you scroll down, we also provide you that ontology, so the classification of my disease. So I've got multiple sclerosis here. It's 3 different subtypes of children and also the different parental terms relative to multiple sclerosis. Each of those boxes are clickable. You can explore the parent term, in this case, nervous system disease, or you can explore the evidence between this gene and that disease. So there is plenty here that you can do from this disease page. And going back to the evidence page here, where the flower can be seen, you can also click on the CD86. So now we are on the target profile page. So you can, get a hint from this page. So it says target. So here I've got all sorts of different types of information, including gene ontology that someone asked earlier, whether or not we have data on gene ontology. We do, so the goal terms are there. So again, you can find out what are the goal terms for this given target of yours. And again, there's plenty of information here that you can explore, including information on the baseline expression coming from GTEx, for example, and all those widgets or visualizations, they are free for you to use in your lab book or your presentation. So please explore this when you have time. And from this target page, what I can do, I can now find out, besides multiple sclerosis, which other diseases are associated with CD86. So we've got that view associations link, and here it's as if I had started from the target-centric workflow. So I search for my target here or from the home page, and I will land on this bubbles view. Again, different things that you can filter by here, whether it's the data type or the therapeutic area that the target is involved. So let's suppose I'm interested in immune system disease, and here I've got my multiple sclerosis there again. I can click on it, again have the flower and explore more if I want to from this flower view. And the larger bubbles here, they represent the therapeutic areas, whereas the smaller bubbles within the larger ones, they are individual diseases. This information is available as a table and also as a tree format as well, that gives you the relationship between the diseases as well. And the different shades of blue, again, they mean the same thing. The stronger the blue, the stronger the association is in terms of our scoring. So we've got around 15 minutes left, so I think we are doing well in terms of time. So the very last demo that I would like to show you, you may have noticed that so far I've searched for one disease. I could also have searched for a target. So let's say if I'm interested in this target called APP. So that's the target-centric approach. And with the web interface as it stands, using this search bar here or going back to the home page, you can search for the targets on a one-by-one basis. So this may not be ideal to some of you. You may have hundreds of targets that you would like to get information from our platform. So that's our third demo that I would like to highlight when you search for several targets at once. So let's suppose that I'm interested in IBD, which is an inflammatory bowel disease. So IBD is one of the diseases within open targets that we are generating new evidence using organoids. So, but from the literature, I've got a list of 26 possible targets, and I would like to see if these targets are represented in other diseases than IBD. And I may also want to find out which pathways are represented in this set of targets. So again, you can search for each of those 26 targets on a one-by-one basis, but clearly this is not the most intelligent use of your time or anyone's time. So we have created the bat search tool, so it can be accessed through the homepage, and this is how it looks like. We've got a sample list here that you may want to load and explore, and I can also paste the targets here one by one if I wish to. Or I may have the list of targets already as a CSV or perhaps as a text file. So you can also upload that list from your desktop, let's say. But in my example, I've got this list here of my 26 targets. I will copy that, and then I will go back to my bat search. I will paste that information. So here we are. And I'll give the list a name. So let's say webinar. I will then load this information. So here we are. It was very, very easy, very quick. And this is loaded now, and you can refer back to this list whenever you can, unless you delete the list from your cache. So the list has been uploaded, and it also has been mapped. So the gene symbols here from my list, they have been mapped to the gene symbols that we've got in Open Targets. So some of those mappings can match exactly 100%, whether sometimes we may have some mismatches. If that happens, we will suggest you what is the best match for your gene symbol. But if you are working on the HGNC symbol, which is the official gene nomenclature for human genes, so you'll be able to get the exact matches from Open Target. So don't worry too much about this. We will do the mapping anyway for you. In this case, everything was matched 100%. And now what is left for me to do is to click on the Analyze Targets. So I've got 26 there, and they will be analyzed now in less than a minute, hopefully, if all goes well. So here we are. So this is the results for my BAT search tool. So I've got 26 targets there. You can explore the results page here. There are several domains of results here. First of all, we tell you which diseases are associated with those targets. So yeah, in addition to IBD, those 26 targets are also represented in other diseases as well, such as Crohn's disease. That representation is sorted as well following this p-value. So the lower the p-value here, the higher the probability that those targets are specific to that disease that I'm looking at here. So actually those targets are more specific to Crohn's disease than to IBD. And from here I can explore the evidence available for those 26, or in this case the 19 targets here. So we can explore that page by opening up this MAD3 gene in that disease. So again, you can see what is out there. Or if you click on this, on Crohn's disease here, you will see actually the 25 targets there associated with Crohn's disease. So again, you can filter as well if you want to. So you can scroll down, and when it comes to pathways, we also give you an enrichment of the pathways that can be seen in that list of targets. So here, 16 out of the 26 targets, they are very much involved in immune system. And we also tell you which 16 targets are those there. If you click on that immune system link, we show you this summary information of the pathway, and this comes from Reactome, and it's very nice that Reactome has now got some of their pathways represented in this very textbook-like cartoon. So again, you can have an idea what this immune system is all about. So you've got the innate part of it, and which cells are involved in that pathway, and so on and so forth. And again, if you want to explore more the pathway information, you can go to the original source. So this is quite nice. If you keep scrolling down from my batch search results, in addition to the diseases that we explored, in addition to the pathways, we also list the drugs that are associated or that can be matched to those 26 targets. So in this example, we've got this drug here that matches ITGA4. And again, if you click on that drug name, you will be redirected to our summary page. From the drug point of view, we tell you whether or not this drug is an antibody, which phase of the trial this drug is currently at. And again, if you want more information from the drug point of view, we link you back to the original source, which is QUICKPOLL. And finally, from the batch search, we also give you an interaction diagram. So we look for information from the OmniPath database. So we display for you this graph that gives you an idea whether or not the targets within that list of 26, whether or not they are interacting among themselves. So here we've got not two, and they do interact with two other targets within that list. So again, this sort of information can be useful when you are trying to prioritize or select a given target for downstream experiments in your drug discovery journey. So I think that's pretty much what I wanted to explore and to focus. I'm sorry that it may have been a bit quicker than you would perhaps have liked. I would just like to finish off before the wrap up actually by saying that the Open Targets platform is not the only way that you can get hold of our data. The platform, the web interface, it's very easy to use, it's very interactive, very intuitive, and we've got amazing, funky visualizations there. But you are more or less restricted with one target or maybe 100 targets if you use the batch search tool. Perhaps you may want to get hold of the entire data sets of Open Targets, so we do provide a data downloads page. So those files are available as JSON files, and we also have a REST API that you can query the data very easily using some of the REST API endpoints. And on that note, I would just like to tell you that if you are interested in programmatic access of Open Targets, please sign up for our webinar on the 27th of November. We can give you more details on that nearer at the time or by email if you wish so. But yeah, in a month's time, we will tell you exactly how to use some of the endpoints in order to get the data programmatically. So in order to wrap up, I would just like to tell you that Open Targets is a consortium that has got two major components. So we've got the Bioinformatics group going about integrating data that's already available out there, integrating that data, making sense of that data and showing it in a very easy to use interface. by allowing this data to be integrated and by displaying this data for free, we will allow you guys to make your own therapeutic hypothesis. And we are using this data integration to make also our own therapeutic hypothesis. So especially our wet lab scientists working at the Sanger and GSK and Biogen, they are using this data to formulate their own hypothesis, and they are generating new data as we speak in one of those, in one of our experimental projects there. And once this data is quality checked, that will be also available for the public, and it will also be integrated in our platform. So that's very much the virtual cycle within the Open Targets Consortium. So the platform itself, it is this place where we have integrated data. It allows you to select a possible target in your drug discovery journey. We also rank the associations based on the different sources. We've got plenty of information when it comes to target and diseases. So you can think of the Open Targets platform as a one-stop shop. And we also make sure that the graphical interface is intuitive and it's easy to use by anyone, whether you are a bioinformatician or not. And these are just some of the numbers of our latest release. And yes, everything is 100% free, and you don't even need to register to get hold of this data. So we believe we are here to support your decision making, whether you work in academia or industry. So there are many questions out there you can use our platform for. If you want to get more information, I would strongly recommend you to read this paper. It was selected as a breakthrough article from Nucleic Acid Research, and it's also a good place. It's the paper to cite us if you use Open Targets in your research. So with that, I would like to acknowledge all the wet lab scientists, the bioinformaticians, also our user communities who've been providing feedback so that we can improve the platform, and also acknowledge the current partners of Open Targets. If you want to know more, there is plenty of channels for you to get support. We've got tutorial videos as well. You can follow us on Twitter, and so there is no shortage of ways to connect with the team. So with that, I would just say that please keep an eye on the upcoming EMBL-EBI webinars. So hopefully it was useful for you guys. Give Open Targets a go. Thank you, and goodbye.